My name is Robert Moody. I happen to be the drug discovery and imaging specialist for the southwest region of the United States. And I'm happy to introduce our two presenters today, Carol Crittenden and Blake Hansen. Carol is an application scientist with Molecular Devices. She's been there 18 years and is the foremost expert on all things Flipper. So she's a wealth of knowledge and is undoubtedly capable of answering any questions that you have. And I think we'll share some pretty interesting data with us today. Along with Carol, we have Blake Hansen. He's the VP of Business Development at StemoniX. He's been there a couple years, and he comes to us from Cellular Dynamics, where he was the Global Director of Business. And so with that, I think I'll thank you all again for joining us and hand the floor over to Carol and Blake. Carol and Blake, the floor is yours.
Hello, my name is Carol Crittenden, and I'm with Molecular Devices. And on behalf of Molecular Devices and StemoniX, we're bringing you a webinar today about using technology, our platforms at Molecular Devices, as well as the cells, the microBrain neurospheroid cells from StemoniX. And what we're going to be doing is drug discovery techniques for phenotypic screening and then compound profiling using structural and functional assays. Today we're going to use the spheroids on both the Flipper Penta high throughput cellular screening system and the Image Express Micro 3D confocal systems to be able to look at the oscillation of calcium within the cells within the neurospheroids, or to look at direct images and staining of the spheroids. And then we're going to talk about multiple parametric evaluation of neurotoxic effects. So just to introduce, and Blake will have a lot more information about this in his part of the talk, but StemoniX microBrain 3D cells are about 600 microns across, and one of these is placed in each well. And they are detectable when they are stained with a calcium-sensitive dye, and when they oscillate calcium, they produce a signal, and that's what you're looking at on the right-hand side of the screen from the Flipper system. And I would also like to introduce the Flipper Penta high throughput cellular screening system, which gives you a choice of camera now, which is a high-speed EMCCD camera, and we also have our standard EMCCD camera. We've upgraded ScreenWorks, and then we also have a greatly upgraded Peak Pro 2 software module to look at characterization of the peaks generated during the calcium oscillations. And we also have increased the images per protocol to 6,000. And this is good for looking at iPSC-derived cardiomyocytes as well as neuronal cell assays. But in addition, we have the same great original assays for GPCRs, membrane potential, potassium, or other assays that are done on the Flipper system. And the benefits of the camera are now we can do a 100 hertz capture of signal, and that's 100 images per second, so that gives a lot more detail. And then we also have on the Penta system the greatly enhanced peak characterization with the Peak Pro 2 software and all of the great technologies we've used before, as I mentioned. So our protocol for the microBrain microspheroids, we receive them in the wells from StemoniX, so they ship them out and they look just fine when they arrive. You culture them for a few days and they're ready to go. The cells are incubated for 2 hours with Flipper Calcium 6 assay kit dye. And then compounds are added and read on the Flipper Penta system at intervals. We started reading in our experiments at about 15 minutes and then up to 24 hours. And the difference in temporal resolution, I can show you here with the cardiomyocyte assay, is that at 8 hertz, you can see peaks, but you can't see the details surrounding the secondary peaks in the peak elongation when treated with compounds that caused this by they're like hurt blockers or whatever. So as you can see with the even with our high-speed camera, just even at 30 hertz, you get a great amount of detail and you can capture the secondary peaks. This is a screen for ScreenWorks 5.0, and you can see all 384 wells on the right. On the left, you can see the detail for one well. You can group them. You can also call up all kinds of assay parameters, and this gives you the ability to capture the peaks, and so you can do like a peak rate or peak height. Also, if you've got characteristic differences in how the peaks are behaving or how the oscillations are behaving, you can capture those. You can capture slopes. You can capture peak widths, heights, rates, all kinds of things. And so the other thing you can capture is an early after depolarization event, which is the width of the peak, and then it'll give another little characteristic peak at the end before the signal drops. You can capture those. And then to take a look at oscillation patterns in neuronal spheroids, you can see the controls, and this is a relative fluorescence intensity of the calcium oscillations over a 10-minute period. And you can see the controls, and then with MK801, you can see variable amplitudes, and it's also slowed down. GABA, which is an antagonist, and it's slowing the beating down as well. And baclofen causes both decreased frequency as well as a spacing irregularity. Here's some more patterns from oscillations in neurospheroids. 4-aminopyridine greatly speeds it up. Valinomycin can slow it down, but it also causes secondary peaks and then also smaller internal peaks. So there's an ability to start to understand the phenotypic differences between the effects that compounds have, whereas you can see chinic acid speeds it up and gives you secondary peaks, and tamoxifen decreases the frequency and gives you variable amplitude as well. Some of the parameters when treated with drug are amenable to concentration dependencies, and you can graph them. Some of them are not, as you can see here as well. So the other thing we did were viability studies using the Image Express microconfocal system, and we characterized the compound effects on morphology and viability of the 3D neurospheroids, and they were treated with 30 micromolar of compound drug for 24 hours. And then the cells were stained with DAPI, which stains the nuclei blue. Calcium AM is a viability stain, and that's green. And then mitotracker orange is a viability stain for mitochondrial cell health, or the health of the mitochondria in the cell, I might say. So you can see you've got a very different pattern of spheroid morphology and viability. You can see each one is very different from the control, versus Taxol, tamoxifen, and then chlorpromazine. And these were also further analyzed, and we used some cell scoring algorithms to generate these images. And again, this work was done by Oksana Sarenko, who is our senior scientist in our group for working with these both the cardiomyocytes as well as the microbrain and other neural spheroids in the group. The other thing you can see is control versus Taxol and tamoxifen, which were two of the compounds you just saw on the screen previous, that showed a difference from control in their calcium oscillation pattern. So you can see there's some correlation between changes. The last thing we did is we put it all together. We took the viability data and then some other, with any place that we had EC50 or IC50 values for peak rate or amplitude. And then we also looked at places where we had oscillation stopping as well as amplitude irregularity, what those concentrations were, to kind of develop a phenotypic profile for each of the compounds. And these were mostly either neurotoxic or neuroactive compounds. So in summary, you can see that it's possible to do multiparametric studies using both the Flipper Penta system and the Imaging Express microconfocal systems. And you can quickly show the effects of neuromediators and neuroactive drugs so they can be studied and detected and then studied further. You can also look at inducers of seizure for their predictive value, or as you'll see in the next talk by Blake, there's neurosensitive, neurotoxic assays can be done, as well as understanding a medical profile. So the other thing we can do with the Flipper Peak Pro 2 software is you can better analyze and have more readouts related to the peak widths and heights and shapes of the curves as far as capturing and predicting neurotoxicity and seizure-reducing effects of compounds. So that's it for me. And now I'm going to pass this on to Blake Anson from StemoniX, and he's going to talk about the science as well as the case studies of using the cells on our systems and molecular devices.
Thanks, Carol, for highlighting a couple of key platforms that we use in interrogating our neural cells, specifically the Flipper Penta system and then the imaging capabilities of the Image Express. We're going to do a little bit of a shift here now and really talk about the wetware rather than the hardware. And specifically is going to be looking at studying neural function in a dish. Now, in vivo, or in the native condition, that neural function is really the result of several upstream developmental and operational processes, be it starting at cell development, into a network formation of neural cells, into that network forming functional neural activity, and ultimately being integrated into the organ. Now, we also know that each of these discrete steps can interact with previous steps as well. Hence, network formation can influence cell development, neural activity can influence network formation, and the integrated organ activity can actually then influence the neural activity, the formation, as well as the cell development. So it's really a bidirectional process that can ultimately be read out at the stage of the integrated organ. That is, anything happens to these earlier processes, you'll see the impact downstream here. So moving from the native condition into the dish, we really follow the same process, but the end result here is an integrated organoid or spheroid composed of multiple different neural cell types. But again, the formation process is very similar, where you have cell development, network formation, the onset and solidification of activity, ultimately being integrated in a functional output from the spheroid. All right. This functional output then can be used to assess things like neurodevelopmental disorders, as well as treatment to identify therapeutic pathways for them. Similarly, it can be used to look at the effects of compound or drug activity on neural function, be that beneficial impact or a negative or toxic impact. And that's really what we're going to talk about today in this half of implementing 3D neural spheroids in drug discovery. What I'll take you through is 3 separate pieces, a very broad basic overview of StemoniX, an overview then of the microBrain 3D. These are human iPSC-derived neural spheroids. And then last but not least, 3 different case studies looking at how the spheroids can be implemented in conjunction with the molecular devices technology to look at toxicity testing, screening as a whole, and then disease modeling. All right, StemoniX is really built on 3 foundational pillars. That is the human organoids, because we believe they provide a richer and more relevant context for better answers. Better answers ultimately lead to better translation. We also then make sure all these organoids are keyed into and used in high throughput drug discovery. This really allows the teams and people to identify novel assets, optimizes the process, and really saves a lot of the cycle time. So saving dollars, FTE, and ultimately giving one more downstream patent life. And last but not least is a series of advanced analytics that moves into machine learning and artificial intelligence. But today, we're going to really focus on the organoids and the high throughput drug discovery aspect. So Carol alluded to the preparation earlier. This is microBrain 3D. It's human iPSC-derived neural spheroid or organoid. It's a co-culture of neurons and astrocytes starting from the same source. So we're not doing an admix, but rather we are co-developing and co-differentiating the cells from neural progenitor cells. So they really have those developmental interactions as well as the mature functional interactions. As you'll see, they exhibit key neuronal and astrocytic markers. There's a robust spontaneous synchronized activity, and it's amenable to higher throughput screening, being in 384 well format. All right, the product itself, as Carol alluded to, is supplied in a microtiter plate, either 96 wells or 384 wells. This example, we're looking at a 384 well plate. Each well has a spheroid. We see a blow up of that micrograph on the right-hand side here, about 600 to 700 microns in diameter. And each well or each spheroid is highly consistent in size. The graph on the bottom left illustrates the mean spheroid diameter plus or minus that standard deviation. So we've got a very high level of reproducibility. These are either shipped to the end user or used in-house in our services. When they're shipped in a hibernated state, not cryopreserved, so the end user is able to use them very quickly after receipt. Simply a couple of media changes, and then you're ready to go. The other advantage here is that although you can use them right away, you can also use them for a long time, meaning that they can still survive for months in culture. We typically have about a month window that we warranty, but users have taken them out several months. So you're able to do both acute and chronic types of exposure. From a characterization standpoint, we're showing here qPCR of looking at some neurotransmitters as well as ion channel expression levels from both the human brain in orange and the 3D cultures in blue. And we can see that although variations exist between the preps, you are seeing the key expression or the expression of key markers, key functional proteins. All right, we have RNA, full transcriptome RNA-seq data that we can mine for you if you have genes of interest that you'd like to know more about. At a cellular level, we can see the key markers here highlighting the presence of astrocytes with GFAP, neurons with MAP2, as well as a key component of synapses, synapsin-1. Altogether, these then demonstrate or come together to form this neural spheroid of neurons and astrocytes with key markers for synaptic function. This culminates then in spontaneous synaptic activity, neuronal activity. We're seeing here an evidence here on the MEA, where we're looking at the activity from 8 electrodes, where a spheroid has been placed on the electrodes and stayed on the electrodes. We can see the raster plot showing synchronized activity, and then the combined plot ahead showing a waveform of spontaneous network-wide activity. All right? Similarly, the neuronal origin of the activity is confirmed and shown here on the right-hand side with a synapsin-targeted calcium fluorophore. So now, whenever we see a light up there, a flash of light, that indicates that the neurons are active. The MEA is a little lower throughput than we would like to turn to a higher throughput system, and that is the flipper. So this again is the image that Carol showed earlier. The spheroids are incubated with a calcium fluorophore, this time in the bath, not just synapsin driven. And the modulation of neuronal activity then is visualized by the flashes of calcium fluorescence that can be visualized on the flipper readout by this oscillatory synchronous activity. This is spontaneous. This is basically the functional phenotypic output of all of those previous stages we talked about: cell development, network formation, and neuronal function. This is our key readout that we'll be talking about for the rest of the program, the rest of the seminar here. We verified and validated the neuronal function here by adding in several different neuronal specific blockers, whether it's ion channel blockers or looking at a variety of neurotransmitter agonists and antagonists. And we can see that in a dose-dependent fashion, we can see impacts on peak count. So that is the number of peaks that occur from the spontaneous activity. So thus, we've got in summary, a microBrain format that's amenable to look at CNS or neural function in the dish. All right. On top of that, to make sure that it's usable or implementable, we want to make sure that we've got reproducibility. And here's a snapshot of that, showing that across 3 different plates, we've got very reproducible spontaneous activity. And then furthermore, in experimental or drug space, chemical space, we see great reproducibility across drug effects as well. Here looking across 2 different experiments, 8 different compounds, the mean result there of the IC50 of each compound, each experiment, shown to fall on top of each other across experiments. So we've got great interplate reproducibility as well as great inter-experimental reproducibility as well. Right? So jumping into the first of the case studies, this overlaps a bit with Carol's talk as well, and was published by her colleague Oksana Sarenko in 2019. Here we're looking at the functional output for toxicity, where you've got your control traces versus the abnormal trace in the presence of a variety of reference compounds or environmental toxins. However, we can also look at biochemical endpoints, again, as Carol pointed out. Here we're looking at viability as well as mitochondrial health. So together, these 2 form a system where you can look at both terminal endpoints as well as functional impacts. Oksana then rank ordered these compounds and compared the functional output versus the terminal output. So the functional output being the multicolored shapes here, looking at that waveform amplitude, decay time, peak width, a variety of different parameters, versus viability shown in the triangles with calcium AM. And as is no surprise to people who do this work, we can see that the functional readouts are much more sensitive and much more amenable to rank ordering toxic effects than simple viability. So we've got really a highly sensitive system that can be used in a multiparametric output. Shifting now to the second case study, we're going to look at basic screening methodology. This graph illustrates the results from screening Select Chem Library, approximately 1,400 compounds, I believe, a single dose, done across 4 different plates. What we're seeing here is that, first of all, we've got a large window, a large assay window. The bottom of it is shown as no activity as brought on by mucimol, a GABA agonist, versus the upper end, an increase in activity brought on by 4-AP. So we've got a wide assay window. Coupled with that is great reproducibility of the DMSO controls shown here in blue across each of the plates. This tight reproducibility leads to a tight assay window shown here in red and defined for this screen as 3 times the standard deviation of those DMSO controls. So now we apply the compounds. The results of seen are illustrated by all the black dots here. Any of those compounds that cause an effect to fall outside of the assay window is considered a hit. That means that compound modulated that particular biology such to the extent that it fell outside of 3 times normal activity. Right? Now, the beauty of the neural spheroid is that you've got many aspects of biology combined together in that neural spheroid. So it's a phenotypic readout, not necessarily a target-based readout. But by using this annotated library, we're able to then determine the different types of biology that one can access here. Now, granted, the majority of the biological hits, as shown by the histograms here, fall within the neural networks, neural transmitters, transmembrane signaling, ion channels. But the long tail of these graphs illustrates that you have access to a lot of other cellular biology as well. This really allows you then, in a phenotypic approach, to really go in and let nature show you the course of your biological investigations, not necessarily coming in with presuppositions. This type of data allowed us then to enter into a research collaboration with the NCATs, part of their NIH HEAL initiative, which is helping to end addiction long term. Basically, looking for substitutes for opioids that may still have the same analgesic effect, but without the addictive effect. The idea here is to be able to identify compounds and really rank them and prioritize them before taking them further into developmental aspects. Right? The last case study is going to be looking at disease modeling. Now, this takes advantage of the iPSC nature of the neural spheroids and draws the biological material from patient populations. This case specifically is Rett syndrome, and in this case, we're looking at an affected male and their unaffected parent. So we've got familial controls here from a patient as well as from a related healthy donor. Now, the disease line we have, again, is Rett. It's from a male. It's a nonsense mutation in MECP2, resulting in a loss of function. Now, the first thing that we did was to generate the lines and ensure that they could be used in the microBrain assay. This is an example looking at a 3D 4-well plate where the rows on the left are the Rett spheroids made from the Rett patients, and the rows on the right, the columns on the right are from the control patient. Again, 1 spheroid per well, a very good reproducibility of spheroid size, and that's quantified and shown over here on the right-hand side. Looking at cellular content, just at a high level with GFAP for astrocytes and MAP2 for neurons, we can see that both spheroid populations have relatively the same organization of different cell types, astrocytes and neurons. So we're not seeing much of a difference there. We're starting to see our first hints at differences in functionality. Here we're looking at neurite outgrowth, where the spheroids were transferred from their 3D 4-well plate into a matrigel substrate, and the neurite outgrowth, is shown here by the fan of neurites coming out of the spheroid, is monitored then over 72 hours. And we can see for the healthy controls, we've got quite an advanced arborization of the neurites, whereas that growth is much more stunted from the patient-derived Rett cells. So we're starting to see the first hints of functional impact of the MECP2 mutation. We then looked at the electrophysiological function, and that's what we focused on. Again, looking at that calcium waveform output. The left-hand side shows data tracings acquired on the flipper from the wild-type spheroids. Again, that nice regular waveform. On the right-hand side, we're looking at example traces from the Rett spheroids. And what we're seeing is that the tracings, the peaks, are more irregular. They're much noisier. Note that the scale bars are the same, and they can also be much more broad. I'm taking this to mean that there's much less regulation of the synaptic activity. Down here in the noise, you've got a lot of neurons firing, but that's not resulting in the whole spheroid being activated like it is in the wild-type spheroid. So you've got really an uncontrolled neuronal signaling here. Each of these different parameters can be quantified and used as the basis for a phenotypic screen. In that screen then, we chose 300 compounds that were picked in conjunction with the Rett syndrome organization. These were compounds that are known to impact pathways that are believed to play a role in Rett syndrome. So the basic mantra or MO of this screen was to take the wild type effect and take the disease Rett phenotype, add compounds, and try and return that Rett phenotype to the healthy phenotype. An example of that is shown on this graph or this slide, where we're looking at peak width, so the width of the of each oscillation. The control phenotype is shown here as 100% in the green triangle. The Rett phenotype is shown up here at 150% in the blue diamond. Again, the average result from a duplicate application of the compounds are shown in black. What we see here is that some compounds, circled in blue, are very good at rescuing this particular phenotype. Other compounds are not very good at all. This gives you a little bit messy data, to say the least. So what we did is took advantage of the ability to pull out multiple parameters of that calcium oscillation, whether it's rise time, peak width, decay time, reproducibility, height, you name it. What we're able to do then is generate a radar plot of those multiple parameters and generate a phenotypic fingerprint, shown here for the wild type. When we then look at that across compounds, we can see, or first of all, across Rett, we can see the striking difference between the control phenotype in yellow and the Rett phenotype in blue. Starting to look at compounds, we can see that some compounds, for example here, procaine, is very good at rescuing one or two of the parameters, width and spacing, but not so good at rescuing other ones, say here the standard deviation or the regularity of the spacing. Whereas additional compounds were even able to affect a greater rescue across multiple endpoints. For example here, compound BIMU8. We showed almost a full rescue across all 7 parameters. All right, so you're really able to differentiate now the level of rescue for each of the different compounds. And given that it's a phenotypic screen, we're not going in thinking that sodium or GABA or HDAC inhibitors are going to be what cures the phenotype. We're letting nature tell us, biology tell us what is curing it, and we can see that we're uncovering then multiple different pathways. So we found hits in serotonin, GABA, AMPA, and just a variety of different compounds, thus giving one multiple shots on goal as to which compounds are taken forward in development. All right, so with that, I'd just like to summarize. I hope I've shown you that the microBrain 3D is a great human-based tool for CNS modeling in vitro. We're able to use it in high throughput. We've got the appropriate biology, and it's really got a lowered workflow burden. You're not doing any of the differentiation, the culturing, or anything. It's really plug and play. They can be used in functional and structural toxicity prioritization, can be used to screen across a diverse number of neurobiological endpoints, and last but definitely not least, can be used to generate diseases in a dish and then screening to find potential therapeutic compounds and pathways. So with that, I say thank you, and we'll take more questions, or we'll take questions at this point. Thank you.
One question is, can large molecules access the microBrain spheroids?
Yeah, they can. Thanks, Robert, for the question. All of the data that we presented here was with small molecules, but we also have a variety of customers using large molecule approaches, including various RNA modalities, ASOs, as well as antibodies. And also we found that the spheroids, even though they are 3D, are amenable to viral infection or transduction.
Great, thank you. Another question came across is there necrosis or cell death at the center portion of the spheroids?
Simple answer there is no, there is not. We have not seen any evidence of that. We have looked by means of examining mitochondrial dyes. So we see that mitochondria are active throughout the spheroid. And then some of our pharma partners have actually submitted the spheroids to their pathology groups where they've done sections and they've seen no evidence of cell death or necrosis either. So we're pretty comfortable that we've got good cell viability throughout.
Thank you. We got one from Mary West up in the Bay Area. Very interesting. She says, can you talk about the bioinformatics end? Are these your own programs, is the question.
In terms of the bioinformatics, we have very limited capabilities with that. We can do qPCR. We can submit for full genome RNA sequencing, and we have that data that we're able to mine for end users if they have a gene of interest, a question about a gene of interest, but we do not necessarily do much of the in-house bioinformatic analysis ourselves, at least not the very sophisticated things.
Yeah, that's helpful. Thank you. I guess that actually dovetails into one other question, and that's whether or not you provide services or just products.
Yeah, a glaring omission on my point from a marketing standpoint. We talked all about product and how it's being used, but we also provide services. And that obviously includes that functional calcium readout that we discussed here in this seminar, but also a variety of other readouts, whether it's, like I said, qPCR, or generating material and shipping material for downstream analysis, such as whole genome transcriptomics, protein analysis. Also, the gamut of biochemical endpoints like viability, mitochondrial function, oxidative stress, you name it. If it can be done in like your standard heterologous 2D culture, we can do it in the 3D culture.
Great. I mean, that begs the question: can microBrain be done in 2D cultures as well?
Oh, yes, it can. It can. We see there with the 2D that the rhythmic oscillation, that function, is not quite as robust, and we've got a publication coming out that details that a little more. So while they're functional, they're not as robust and as tight as the 3D, which we take to mean that the 3D has a little better or more mature, if you will, functionality. We do see a lot of utility with the 2D for transduction, transfection, and imaging, although we and some of our partners have become quite good at imaging the 3D as well.
Good to know. Good to know. I think you may have addressed this to some extent earlier, but I do want to be respectful of all the questions coming in the Q&A panel. So is artificial intelligence data mining built into the software?
Not built into the software, but we are able to pull out quite a bit of granularity and robust features across the phenotypic output, and that is then ready for the leap into AI and machine learning. And we're doing that both from a toxicity standpoint as well as a phenotypic rescue of disease lines.
That makes sense. Thank you for the clarification. And one more here.
Quick too.
Oh, please.
Yeah, this as far as getting ready for that AI, the Peak Pro 2 software coming off of Flipper and doing the analysis on the calcium oscillations is a module that we have that goes along with the software that runs the Flipper Penta. And that's where you're getting the peak heights, rates, peak widths, and that kind of stuff.
Fantastic. And our question asker says, thanks. One last thing. Got a question for something a little topical. Given the recent publications on COVID-19, is microBrain being used in these types of studies?
Yeah, we started some early studies, very preliminary, about hearing the off-target meaning non-respiratory effects of the SARS-2 virus. And so we've done some studies in collaboration with UCSD investigators, because we do not have a BSL-3 lab, but we have preliminary information that does support infection or infectivity of both the microBrain as well as microheart preparations. And we think, I would say we're excited, which seems to be a little odd to say about COVID. I don't mean it that way. We are excited about the fact that we see this infectivity in a higher throughput screening format. So I think this is going to provide really a way of ameliorating or preventing potentially some of these effects we see in other tissues rather than just simply the lung tissue.
Yeah, that explains a lot. Thank you. Well, we'll see if that's the questions that I have for the moment. If anything else pops up, any of you attendees, go ahead and pop something in the Q&A panel. We'll address it. But as we await any further questions, I do want to thank everybody for attending. I want to thank our speakers, Carol and Blake, for their time and for the effort that's put into generating these data. Very interesting indeed. And I wanted to remind the audience, as we're looking at these email addresses, that if you have any Image Express, imaging, or Flipper-based questions, please feel free to reach out to either me or Carol. And if you have any questions about microBrain or all things StemoniX, please reach out to Blake. And I do also want to let everybody know that if we have, as we have recorded this seminar, we will have a higher resolution video ready, and we'll be providing that to you via email in an on-demand version so that you can review things and share with others. We'll check one more time. And, yes, Mary says she would love a copy of the presentation. Perfect. We'll make sure we get that for you. One other question here, just coming in late. The spheroids, while shipping, are the effects of hypoxia, stress, or phenotypic changes from the environmental stress during shipping? Have you durability tested your shipping regime? I guess
yeah, definitely. And we do not see any negative effects. You know, in fact, the pathology study was done with a pharma partner off-site where we shipped. We're able to ship the microBrain plates both domestically and internationally without any problems. You know, we've got customers in both Europe and Japan. And we recently have a fairly new shipping process that makes it even easier and smoother, if you will, for customers to unpack them. Basic process is you're able to peel back, you know, a cat mat that we have, shipped at ambient temperature, do a media change the day that you get them, and then a couple of more media changes, and then they're ready to go.
Great. I guess, yeah, next step will be shipping them via drones, right, Blake?
Yeah, there we go.
All right. Well, I think we've got some good questions. And, here's another. What is the method of aggregation for the StemoniX spheroids and the microBrain? Gravity-based, Matrigel, et cetera.
Yeah, so we really, as part of our proprietary methodologies, mainly in the cooking sauce, but they're formed in suspension. So it's not really a gravity-driven, but there definitely is not. It's not a matrix-driven either.
Got it. And I appreciate that we obviously can only share so much.
Right.
But no, an important question that was buried into rather than in the Q&A panel, it was in the messages. So thank you for bringing that to my attention, Shelley. And I think that might do it. Let me take the opportunity to thank our speakers again, as well as the attendees, and remind you that these email addresses can be used to talk, to get information about imaging or Flipper to Carol and Robert, myself. And then anything to do with microBrain and StemoniX can be forwarded to Blake. And with that, you'll look for an email from us with a link to the high-res video. And I thank you again. And everybody have a great rest of the week.
All right. Thank you, Robert. And thanks, everybody, for attending.